Hello, my name is Michael West. Few scientists have played such a fundamental role in the history of aging research as Dr. Leonard Hayflick. In the early 1960s, Dr. Hayflick reported for the first time that human cells age when cultured in a laboratory dish. And so this became a fundamentally new way of studying and understanding human aging. In November 2016, I sat down with Dr. Hayflick for an afternoon of discussions talking about the history of the field and his experiences in it, and then looking forward to the future of the understanding of human aging. Here in 4 parts are my conversations with Dr. Hayflick. So Len, many of us think of you as the father of modern aging research. Why do we think that?
Well, I don't know how accurate that is, because when I entered the field, I actually entered it yelling and screaming, not knowing anything about it, and after knowing a bit about it, realizing that there were other fathers who preceded me.
Right.
Anyhow, I accept that as a compliment, and I appreciate it very much. I think the reason for it is my discovery made in the early 1960s that overturned a dogma in the field of cell culture and in cell biology that was believed since about the year 1900. And that belief was the following: that if you put cells in culture, that is into laboratory vessels, at that time glassware, today plasticware, that the cells will divide and continue to divide as you pass them from vessel to vessel as they grow further and further or reproduce further and further, that the cells will divide indefinitely. They're immortal, in other words.
Right.
Well, that belief, when appreciated by people in the field of the study of the biology of aging, meant that if you can take tissue from a tiny bit of tissue as big as a pinhead from any part of your body and put it in culture and find that it will divide forever, has an important implication, and that is that aging is not an event that occurs in individual cells, and so it must have some other cause, which was a major interest in biology for many, many years. That is the cause of aging. So my discovery that was wrong, wherein I discovered that normal human cells have a finite capacity to replicate, overthrew that dogma, and I interpreted my discovery to be telling us something about aging, that aging now has to be thought of as an intracellular event, an event that occurs inside of cells and not outside of cells. At that time, it was believed that aging might be caused by radiation or stress or any other cause that you might invoke that was other than based within a cell. So that I would guess that might be the reason I've been anointed with this name, Father of Study of Aging, because that changed the focus of the field completely.
Well, after all, we're made of cells, and if cells are aging, it's pretty fundamentally important in human aging. Well, let's go back in time to try to understand how all this evolved. So, why don't you just tell us briefly about, where you grew up and how your interest in science began?
Well, I was born and raised in a row house in southwest Philadelphia, noted for houses that are adjoined, have a common wall, and go for a city block in one direction. And my parents were in average, if low average, financial circumstances as a result of the Depression. And having been born in the year 1928, a year after the big crash, and being raised in subsequent years in the early 30s with parents who were essentially poor, And I remember many examples of this. My father, for example, couldn't afford the 15 cents necessary to ride trolley cars from Southwest Philadelphia to the center of the city where he worked. And so he walked that distance. It was about 10 or 15 miles in each direction, I think. He wore out his shoes and saved them as long as he could by forcing newspapers into the soles of the shoes in order to keep from walking directly on the pavement. That's just one of many examples. He was not alone in doing things like that during this period. Later on, as people began to earn a few more dollars, I became very interested in chemistry when an uncle of mine bought for me what was then called a chemistry set. The manufacturer was Gilbert. It was a very popular company at that time that made chemistry sets and small engineering sets for kids. That got me started. I should say parenthetically that all of the chemicals that were in that set at that time are now forbidden to be used by children and even by adults because some of them were dangerous if not used properly. So my interest got stimulated to the point that I asked my mother whether I could build a laboratory in the basement of our house. And being a very liberal lady, she agreed, and I set about buying construction materials and building a pretty nice lab in the basement of my house with shelving and bottles containing all kinds of chemicals and doing experiments at that time. The most interesting to me at that time were rockets and explosives. I even went to the extent of having discovered at a chemical supply house near the University of Pennsylvania that they had stored there elemental sodium, which is a very dangerous material. It's kept under kerosene because it's highly reactive with oxygen and will explode in the presence of oxygen. And I think I was maybe 12 or 13 or 14 years old at the time. And this fellow very wisely said, "No, I can't sell you that because I would need a letter from your mother." I went to my mother with the problem and she said, "Sure, I'll sign a letter." This is how liberal she was, and also in depending on my intelligence not to do anything very dangerous, because she didn't know what elemental sodium was, nor did she know what any of the other chemicals I was working on could do. So I obtained this material and constructed a gadget in which a cube of sodium was tied with a string while my hands and all of the things that touched it were dipped in kerosene. And the cube of sodium was tied to the end of a string, and the string run through the unopened end of a can where we punched a hole. And the string was then pulled, and the sodium cube was then raised to the ceiling of the can. And the mouth of the can was put into a dish of water outside, of course, in our alleyway. And the string was about 15 feet long, and so if you let go of the string, the sodium block would fall into the water and explode and send the can like a rocket up into the air. Those are the kinds of things we did. I won't bore you with all of the things involving rocketry and flares and how to produce different colors and things of that sort. Well, I began to learn so much about chemistry that when I got to high school, and I had built up quite a collection of different chemicals which fascinated me and the things that could be done with them, that in my first year chemistry course in high school, I found that the teacher, who I liked very much, he was a very nice man, had made some errors, and I made the mistake of trying to correct him in public, that is in earshot of the rest of the class, which was not a good thing to do. However, it allowed me to be told by him that from now on you're going to help me in the stock room. I don't want you to sit in on my lectures anymore. An annoyance for me. That was a big mistake because in the stock room were chemicals that I didn't have, and so I could put a few ounces of those into a piece of paper and take them home with me and build up my chemistry library. But he was a very nice fellow, and I was taught a lot by him. And similar things happened with teachers of physics, etc. When I graduated high school and ultimately ended up at the University of Pennsylvania as a student, I was not sure what I wanted to major in, chemistry being one of them, but I then became very much interested in microbiology. I decided to take a course in microbiology and got fired up when a very unusual thing happened. I was sitting in class on, I think, the first or second day of class, and after the lecture, introductory lecture, we all filed into the laboratory, took our places, and as I was sitting there, the laboratory technician, a young woman, walked by with a tray of test tubes, and each test tube, as I later learned, had what's called a slant of agar, a kind of gelatinous material on which bacteria can grow. And the tray that she was carrying had about 20 or 30 of these tubes, and each one had a different color. And I was astonished to learn later that each microorganism gave a different color because of its biochemistry. Well, that, for inexplicable reasons, but the fascination with the coloring, turned me on to microbiology, and I became very interested in bacteriology and other biological studies like parasitology, etc. And graduated from Penn with a bachelor's degree. The professor of microbiology turned me on to a job that was available at what was then Merck Sharp and Dome, or rather I should say Sharp and Dome, which was taken over by Merck soon after I was employed, which I knew about at the time of employment. And we moved up to from Glen Olden, Pennsylvania, to another town outside of Pennsylvania. And that was my first job, to work in a biological laboratory, microbiological laboratory at Merck.
I see. So your beginning of moving from chemistry to biology, if you're at all like me, I mean, it was chemistry shows us the power of science in a way. Mm-hmm. If you can send a rocket into space or you can cause an explosion, by understanding chemistry, there's power in that knowledge. Was it partly perhaps your fascination with the growth of bacteria that, maybe there's power in understanding life?
Yes, I think that's a fair observation. It became apparent to me, of course, early on that chemistry was the basis of all biological reactions in respect to how one defines terms, so that it became of very great importance in respect to background to have this knowledge of fundamental chemistry and organic chemistry, as it later turned out I took courses in. And that became the basis of my interest in microbiology. And I should say that microbiology, the aspects of microbiology, at least in the 1950s and 1960s, could be lifted entirely over to the then emerging field of cell culture, where cells are cultured under the same circumstances and the same general technology that one uses in microbiology. So my microbiology training stood me well in respect to the events that occurred later with my work in cell culture.
I happen to have a book here, which is Theodor Schwann and Schleiden's first announcing the hypothesis that life is made of cells.
Schleiden's wrong, yeah.
And here are, in the back, are the, the plates showing Schleiden's pictures of cells in plants and Schwann's pictures of cells in the case of animals. What is a cell biologist? It's hard to explain maybe to all audiences, but what fascinates me is the fact that we're made of cells. We are a multicellular organism. Putting human cells in particular in the dish and peering down at them, you sort of have them trapped, you're in my territory, you're in the world of science. We've taken you out of the body and we have you now, we can study you. And putting human cells under the microscope and looking at them, I get that same mystery and wonder. How do these cells work? And how does the aging of cells work? Did that intrigue, did that mystery play any role in your gravitating from your early research and interest in chemistry and microbiology into human cell culture? How did that interest begin with you?
Well, it actually occurred under less sophisticated and less logical pathway than you described. It occurred because my mentor at the University of Pennsylvania, during the time that I was there as a graduate student, where I went from Merck to graduate school at Penn, he had just returned from the first course in cell culture given at, I can even remember the name, Mary Imogene Bassett Hospital in Cooperstown, New York, where the first course was given by the major personalities in the field at the time, and there were only about half a dozen or fewer. He came back full of enthusiasm about this new, what was thought by him to be a new discipline. Actually, it wasn't. And I was working with a group of microorganisms called the mycoplasmas, now called the mycoplasmas, then called PPLO, the smallest free-living microorganisms. And he was so full of enthusiasm about the course he just returned from, he tried to convince me to do my Ph.D. thesis on cell culture. And I resisted for quite some time until I realized that this assistant professor was my mentor and I should listen to him more directly. And I decided, with his approval, to do a PhD dissertation on the growth of mycoplasmas in cell culture. So I combined the two and built a laboratory along with another graduate student in the laboratory with me. And that got me into the field of cell culture in particular. From there, I moved, after my graduation with a PhD, to the laboratory of one of the most famous cell culturists in the country, if not the world, a man by the name of Charles Pomerad, whose name is not very well known today, but at that time it was very well known. He attracted cell biologists from all over the world to his lab in Texas, the University of Texas in Galveston, where I went, and studied there under his tutelage for a couple of years.
What years would that be?
That would be 1956 and 7, because in 1958 I returned to the Wistar Institute where I had done my master's degree work. And so I was essentially returning home in a sense, because the Institute at the time of my return was taken over by a new director. He was full of enthusiasm, did a lot to expand the Institute. His name was Hilary Kaprowski. I should say a word or two about the Institute itself. It was the first biological research institute in the United States, founded by the Wistar family in the late 1800s. It was called technically the Wistar Institute of Anatomy and Biology, which is the clear name for it. It's a standalone institute with very loose affiliation with the University of Pennsylvania. It is not a part of the University of Pennsylvania technically. Kaprowski, who was a virologist and was a pioneer in the development of attenuated poliovirus vaccines, Kaprowski was the director, as I say. He came from Lederle Laboratories. And because of his international reputation in biology, and especially in virology, the Institute began to attract many of the well-known personalities in the field of cell biology, cytogenetics, chromosome biology, virology, bacteriology. A couple of the advantages that brought people to the Institute as visitors was the fact that we had an apartment in which they could stay, a very nice apartment, which at that time was a bit unusual. It's not today. And also Kaprowski's personality. He was a very gracious host, although there is another side to his character that was not as welcoming. And he entertained on a large scale, and this reputation went worldwide. He traveled widely. He spoke several languages. He was Polish by origin, spoke several languages. And he was also a concert level pianist. And all of these things taken together made the Institute a kind of Grand Central Station, crossroads for biologists of many disciplines, many of whom I met because of those facts. While at the Institute, I was hired to establish a cell culture laboratory, and at that time to provide cell cultures to the other senior members of the Institute, of which I was not one. I took the job because I knew from experience and discussions with other people that by taking the job of a service laboratory to provide people with cultures, that once I had that set up, I could then do what I wanted to do in respect to research and not have to necessarily apply for grants and go through the struggle of grant applications. I could work in this laboratory, do what I wanted with cell cultures, and not be burdened by that necessity. And that's in fact what happened. It worked out perfectly for me. Once I got the laboratory set up in about a year or less, with technicians providing all the necessary services, I then was free to do what I wanted. And one of the things I wanted to do at that time, based on what was then being learned, and that was that viruses seem to be implicated in the cancers of lower animals, in particular rodents like rats and mice and chickens, that the likelihood that viruses might be the cause of some human cancers seemed like a reasonable expectation. And so I decided to look into that question with a very primitive design, experimental design. What I thought I would do is culture human tumors, and the tissue I could easily get from across the street at the University of Pennsylvania Hospital from surgeons who did that kind of cancer removal. And I did indeed, as I recall, culture about 500 human tumor specimens over a period of a year or two. And then what my intent was is to take the fluids in which these cancer cells were growing and put those fluids onto cultures of normal human cells. Well, that presented a problem at that time because it came to be known that first of all, getting normal human tissue is not easy. People don't surrender their normal tissue very easily. And also the fact that adult human tissue was known at that time to be housing unwanted normal viruses like chicken pox or maybe even polio, and I didn't want my system to be burdened with these unwanted viruses if they should spring up during the course of my work. It was also appreciated that human embryos or fetuses did not contain these viruses, that they somehow are acquired later in the life of a human. And so I focused on getting tissue from human fetuses. I did obtain many from the University of Pennsylvania, later from Sweden, as a result of a gentleman who worked in Stockholm and was a visitor at the Wistar Institute with whom we made contact. So during the course of culturing these normal human cells, I, of course, received knowledge that a human fetus was available from a surgical abortion at that time at random times. The phone would ring and a technician would go across the street and get the tissue and bring it back to my lab. Most of the time the telephone would ring at 4:45 on Friday afternoon. But we would not miss any opportunity to get this tissue, which was not available often. But that was an important point, the fact that the tissue that we obtained was obtained at random times, and I'll explain why that was critical in a minute. So I cultured the cells from the tissue that we obtained from these fetuses, human fetuses, and the tissue that I chose to use was lung tissue, mainly because after 2 or 3 months of gestation, the lung is fairly well formed and easily discerned and easily cultured. I later cultured many other tissues, but I'll save some time by just saying that I focused ultimately only on lung tissue and what occurred by using that tissue. So the cells that grow out from the lung tissue at that point in the genesis of the human embryo are fibroblasts. These are a structural cell found in virtually all tissues and considered at that time, and even to this day wrongly, I think, as a kind of weeds. They're not weeds, they have very important functions. In the course of culturing these cells, what would happen is that you would isolate millions of cells from a single piece of tissue, put them into a culture vessel, and in media in which they grow. The cells would then drop down due to gravity onto the floor of a culture vessel. And the culture vessel is then stoppered and put into an incubator so that body temperature of 98.6 degrees Fahrenheit, 37 degrees Celsius is maintained, in which the cells wish to grow best. And then after the cells replicate on the floor of the culture vessel, they will replicate to the point where they fill the entire floor. And if you want them to replicate further, you have to remove them, which is easy to do. You pour off the media, the cells are stuck to the floor. There's a chemical that's used to release them called trypsin. And the released cells are then, for example, and I'll use a simple example to make it easier to understand, the cells would then be divided into 2 equal portions, put into 2 daughter vessels. You then put those vessels in the incubator, wait 3 or 4 days. The cells then reach confluency, as we say, which means they cover the entire floor of the vessel. Now it becomes necessary to do this trick again, and you do that every 4 or 5 days until the cells are, bottles increase by powers of 2. As you can imagine, in several weeks you're going to be up to your neck in bottles unless you do the following, which we did routinely, and that is to freeze the cells. You can actually take normal human cells, and others as well, and by a known technology, put them in a freezer where they will remain alive, or at least alive in the sense that they're static in the freezer, but will take off after you reconstitute them and continue their replication. What I found was this strange event. We had what was called a walk-in incubator at that time because so many people in the Wistar Institute were using cell cultures. We had an actual incubator that was a room held at the temperature that I described earlier. And each person was assigned a certain shelf or two. And I used to go in every day or two to look at my cultures. By this time, I had a half a dozen or more cultures from different fetuses received at different times. I walked into the into the incubator room one day, and I could tell the condition of the cells by holding the bottle up to a light that was hanging in the room and get a good idea of how well they were doing. And one day I walked in and one of the cultures didn't look very well, took it out, put it under the microscope. Sure enough, the cells looked in bad shape, very unhappy. They seemed to have stopped dividing. And I thought that this was probably an anomaly, that the technicians working on them contaminated them maybe with a microorganism or a virus, or made some technical error that I couldn't explain. And so I didn't take this first observation too seriously. As the weeks went by, more cultures began to show the same condition, that is, cessation of cell division, of replication. They couldn't be divided any further, or subcultured any further. And then I did the obvious thing and went back and looked at my notebook and realized this strange observation, that the cultures that had stopped dividing were the ones that were the oldest. They were the ones that were received 8 or 9 months ago. And unlike the cultures that were set a month or 2 or 3 ago, which were, those were perfectly happy, the ones that were 8 or 9 months in culture had been the ones that were showing the cessation of division. Well, that became very interesting to me, I thought, because no one had ever observed this before, and I was either making some blunder, or my technicians were making some blunder, or this was a real observation. By this time I had joined with my very good colleague Paul Moorhead, who is a cytogeneticist, that is a person who looks at chromosomes. And I decided that we should do the following experiment. The experiment was very simple to understand, based on Paul's ability to distinguish between male and female cells. Now one is not wearing blue booties and the other pink booties. What's happening is that you can look at the chromosomes and tell XY chromosomes, female chromosomes from double X chromosomes for the other gender. So you can make this distinction by cytogenetics. Well, that was a key marker in doing the following experiment. Based on my then understanding that the cells underwent about 50 population doublings before they decided not to divide any further, we took a male culture at about the 40th population doubling and mixed an equal number of those cells with a similar number of female cells at, let's say, the 20th doubling.
Now, you're describing the dirty old man experiment? Isn't
That's what we called it later, the dirty old man experiment.
Mixing an old man's cells with
a young lady's cells.
Isn't it more than that? Wasn't your secretary, I heard, protesting?
Yes. We had
Those were different days.
Secretary who, like most people at that time, didn't appreciate the subtlety of distinguishing male and female cells on the basis of chromosomes and no other criterion. And she objected to the use of her cells for that purpose, so we had to go to some other lady who was more knowledgeable about the immorality of this experiment. In fact, we did the reverse experiment later, which is a dirty old lady experiment. So both genders had their say in this experiment. And the experiment initially with the older male cells and the younger female cells, after the mixture went through a number of subcultivations with the unmixed male and female cultures kept as controls, we noticed that the male culture, the control, the cells in that control bottle or bottles had stopped dividing, and the female controls were still dividing. Then we looked at the mixture. Sure enough, the only cells left in the mixture were female cells, which coincided with what we had seen in the two control cultures.
So what that implied was whatever was causing this aging of the cells was somehow intrinsic to the cell itself. It wasn't just
yes.
the media being bad or exhausted.
Yes. Yes, it was hard to imagine a condition that is contamination, technical problem, phases of the moon, whatever you might want to invoke, that would distinguish between male and female cells. It just didn't make sense.
What year was that done?
That was done in 1959 or 60, because the paper that we published was published in 61. So those years were the probable years in which I made this observation. And that was a key experiment because it came close to proof, actually, that the cause of the cessation of division in normal human cells was an intracellular phenomenon and not based on some extracellular event, like contamination or whatever else you want to invoke.
Well, Len, the science is always all about, peer review and presenting things in meetings with other scientists. How did scientists at the Wistar Institute first respond to your thought that human cells might age? I mean, was it seen with skepticism, or did people say, well, this is really interesting and important? Or?
Well, like most major advances in science, it was not believed. It was thought that there must be some fundamental error, because after all, I was arguing that the dogma that was held by senior cell biologists for 60 years was incorrect.
Let me refresh your memory on something. Let's go back in a time machine. I'm holding here Scientific American, 1936, okay? Go back in a time machine and tell me what you think about this. So this is...
You're showing me a picture of Alexis Carrel.
Alexis Carrel. So, Scientific American, chicken hearts, isn't he the scientist that has kept a chicken heart alive for 10 years? That's what the average man quickly links with Dr. Carrel. But the truth is that Dr. Carrel has kept a chicken heart not only alive, but beating in a glass tube if the nutrient solution and constantly growing for 24 years, 3 months. This small, famous portion of chicken giblets seems to be well on its way to immortality, both in a literary and a literal sense. He was the first American, I believe, to win the Nobel Prize in medicine. Isn't that correct?
That might be correct. I didn't realize that he was the first American.
I think so.
Was he an American citizen, actually?
Well, the first person maybe living in America or working in America.
Yeah.
But he was at the Rockefeller Institute, which
correct.
was then this very prestigious institute. Peyton Rouse was the one that did the early work on chicken tumors and everything there. This had to play in a role into that consensus.
It played a very important role in my work, and I'll explain exactly what role it played and how it was ultimately adjudicated.